Hello everyone, welcome to Pharma Icon, and the today's topic is about the important subject in the Bachelor of Pharmacy, that is Pharmaceutical Microbiology. What is microbiology and why we need to study these pharmaceutical microbiology? Okay, first of all, what is microbiology? The microbiology, which is mostly deals with the we need to study about the microbes like microorganisms, bacteria, fungi, and other types of microorganisms, along with their detailed study about their structure and how they are useful, and some are harmful bacteria, and why we need to study. Okay, and examples of some of the microbes are, they are mostly very useful microorganisms are there, and they are very important for the life on the earth. First of all, introduction about the microbiology and history about the introduction and history of the microbiology, and the coming to the history of microbiology and the history and the ancient onwards, the microbiology inventions are occurred in this. The scientist works under this category of microbiological studies are mostly Aristotle. He mostly invent the existence of microorganisms along with Robert Hooke and Antony van Leeuwenhoek. The Antony van Leeuwenhoek is called as a father of microbiology. The Antony van Leeuwenhoek, he is the father of the microbiology. Antony van Leeuwenhoek, he is the father of microbiology. And the Louis Pasteur. Louis Pasteur is one of the scientists who also involved in the microbiological studies. And the next one, what are the branches and scope of its importance? Okay, what are the branches coming under the microbiology? Some of the branches including in the microbiology, they are mostly bacteriology, mycology, phycology, protozoology, virology, nematology, and aquatic microbiology and aerobic microbiology, along with parasitology. And other important categories are microbial ecology. Okay, these are the different branches are mostly involved in the microbiology. And what is the scope and its importance? The scope is nothing but mostly we need to study about the what are useful microorganisms and what are harmful microorganisms, and how they will be used to us. Okay, next, introduction to the prokaryotes and eukaryotes. What are called prokaryotes? The prokaryotes are nothing but type of organisms in which there is no nucleus. Okay, there is no nucleus are called prokaryotes, while eukaryotes are in which the organisms are consisting of the nucleus with the cell membrane, like cell organelles containing bound membrane cell organelles. Like what are cell organelles? The cell organelles like mitochondria, ribosomes, plastids, endoplasmic reticulum, Golgi apparatus. So in eukaryotes it contain different type of cell organelles. Next, the fourth topic in the first unit is study of ultrastructure and morphological classification of bacteria. Okay, how we need to identify the bacteria based on its structure. Okay, first of all, what are the membranes? Some bacteria containing membranes. Okay, and first we need to know about the what is bacteria. The bacteria is a type of organism which is mostly single celled organism is present entire the earth. Okay, this is called bacteria. And how many types of bacteria are there? Useful bacteria and harmful bacteria. Examples of useful bacteria are what? The examples of useful bacteria like E. coli, Rhizobium, and Lactobacillus. Where is E. coli is used? The E. coli is the type of bacteria which is mostly present in the our intestine. Okay, and next, what is Rhizobium? Rhizobium is a type of plant bacteria which is mostly present in the plant, Azotobacter rhizobium. And what is lactobacillus? The lactobacillus is mostly used for the process of making of curd by using the milk. This is about the useful bacteria. And what are harmful bacteria? The harmful bacteria are mostly type of bacteria. They are usually causes diseases to our body. Examples for harmful bacteria are Salmonella typhi, which is caused typhoid, and Mycobacterium tuberculosis. It is a type of bacteria causes tuberculosis. This is about the useful bacteria and harmful bacteria. What are the nutritional requirements for the bacteria? The nutritional requirements using the bacteria are mostly water, carbon, nitrogen, sulfur, and inorganic salts along with the growth factor. These are mostly nutritional requirements used for the growth of the bacteria. And what are the raw materials? The raw materials are mostly used to grow the bacteria include their agar, peptones, inorganic salts along with the carbohydrates and thioglycolic acid. These are the raw materials used for the growth of the bacteria. And why we used these culture media along with the physical properties for the growth of the bacteria. And the fourth topic is growth curve. First of all, what is growth curve and why we need to study in this bacteria? Growth curve is a type of a parameter in which the graph shows the number of bacteria. Growth means it is a type of a feature bacteria, whether it is live or not, we can easily identify by using the growth curve. Okay, it is indicates the response. What is the response of the bacteria, whether it is growing or not. And the next, isolation and preservation methods for the pure culture. What are the isolation methods we need to use for the maintenance of the pure cultures? And the next topic is cultivation of anaerobes. What is meaning of anaerobes? Anaerobes are nothing but these are the type of organisms. They are mostly grown in absence of oxygen. These are called anaerobes. Okay, oxygen free environment they mostly used. And what is the cultivation methods involved in the anaerobes? They are mostly the cultivation media is of anaerobic chamber and oxygen control, and next anaerobic agar we need to use it for the cultivation of the anaerobes. Next, what is a quantitative measurement of bacterial growth? The bacterial growth is mostly of two types, total growth and the viable count. Total count and the viable count. What is the meaning of total count and the viable count? And the total count is mostly the total number of microorganisms present in the given sample. And the what is a viable count? And the viable count is the number of organisms to be living in the given sample is called as a viable count. Viable means number of living. The second topic is the study of different types of phase contrast microscopy. What is meaning of phase contrast microscopy? The phase contrast microscopy is a type of microscopic studies in which we are used to study the different types of cells, tissues and microorganisms. The principle involved in the phase contrast microscopy is the object can be easily illuminated by using of the phase. The one phase is involved to giving the individual microscopic organism structure. Okay, that is called phase contrast microscopy. And what is a dark field microscopy? Here name itself we can indicate it, the dark field. There will be a dark background will be there in that microscopy to easily giving the identification of the original object. Okay, we can easily find out the object by the dark background. Okay, and what is a electron microscopy? The electron microscopy in which we use it to illuminate the object in the presence of an electric beam. Okay, electrons, a beam of electrons will be transferred. By the presence of that electrons we can easily identify the object. Okay, this is about the topics covered under the unit 1. And the next, the unit 2, what are the topics we need to discuss in the unit 2 are, the first one is a identification of bacteria, mostly using staining techniques. Okay, we in the first unit we will study about the types of bacteria and culturing techniques of the bacteria, and in the second unit we need to study about the staining techniques of the bacteria. First of all, how to identify the bacteria. Identification is one of the biochemical tests in which we need to identify the bacteria mostly by the gene sequencing. How many types of genes are present and how we will differentiate that. And the other studies like NMR studies and the cultural techniques. Okay, what are the staining techniques mostly used? They are the type of staining techniques mostly involved by using of stains or different type of dyes. By using the different type of dyes we can easily identify the bacteria. So how many types of bacteria are there? Simple bacteria, gram positive bacteria, gram negative bacteria, and acid fast bacteria. Okay, these are the different types of staining techniques. These staining techniques are also called as a biochemical tests. Okay, and what are the meaning of biochemical test? The biochemical tests are biological procedures can be followed by using of the chemicals. Okay, this is called biochemical test which is mostly used to identify the bacteria and find out how much levels of enzymes are present in the bacteria. This is about the biochemical test. And what is the meaning of IMVIC? The IMVIC, in which 4 tests we need to identify the bacteria. The first one, I means what? Indole test, and M means what? Methyl red test, and V means Voges test, and other C means citrate test. These are the 4 tests to be involved for the identification of bacteria in the staining techniques. And what is a study and principle procedure mostly involved in the methods of sterilization? First of all, what is sterilization? Sterilization is a technique in which mostly used to identification of the bacteria and in which to prevent the growth of the microorganisms. Sterilization means killing the growth of the bacteria is called as a sterilization. Methods to be followed in the sterilization are mostly dry heat sterilization, moist heat sterilization. These are mostly comes under the heating techniques, and the remaining are the radiation sterilization, gas sterilization, chemical sterilization, etc. And the other sterilization methods are mostly physical method of sterilization, steam sterilization, dry heat sterilization, boiling or water radiation. And the chemical methods of sterilization are mostly involving the using of chemicals like ethylene oxide, alcohol, hydrogen peroxide, and glutaraldehyde. These are the different types of chemicals used in the chemical method of sterilization. And physical method of sterilization mostly involving the using of heat and radiation. And what are the mechanical methods of sterilization? The mechanical methods of sterilization like filtration, scrubbing, and washing. These are the mechanical methods of sterilization. What are the evaluations of efficacy of sterilization methods? These are how we need to evaluate the sterilization methods. First of all, the following tests are involved in the evaluation of the sterilization methods. They are mostly bioburden test, and the second one is biological indicators and endotoxin test along with the chemical indicators. So these are the different types of evaluation methods we need to follow the sterilization of the bacteria. Okay, and this is about the different types of sterilization methods and equipments employed in the large scale sterilization. What is large scale sterilization? Large scale sterilization means other than the laboratory conditions. Okay, what are the equipments used in the large scale sterilization? They are mostly autoclave and alcohols, chemicals, and quaternary ammonium compounds along with peracetic acid and electron beam sterilizers, dry heat sterilizers, and ethylene oxide sterilization along with the steam sterilization. These are the different equipments mostly used for the sterilization in the large scale method. And the last topic is about the sterility of indicators. What is the sterility indicators? Sterility indicators are type of indicators which are mostly used in the process of sterilization. These indicators are mostly monitor and check the quality of the process in the sterilization. Okay, in the sterility indicators they are mostly using of the biological indicators, chemical indicators, physical indicators. And the example for the biological indicators are the self-sustained biological indicators, and chemical indicators like using of acids and other chemicals, and physical indicators like temperature and pressure. These are act as an sterility indicators. So this is about the topics covered under the unit 2. And the next is about the unit 3 topics. In unit 3 we are mostly deals with the study of morphology. First of all, what is morphology? Morpho means structural or analytical changes. Okay, the morphology is mostly deals with the size, shape and structure of the bacteria and any other types of microorganisms. The morphology of bacteria is mostly of Diplococcus, Streptococcus, Staphylococcus, etc., in which the arrangement of group of organisms. Okay, next, classification, reproduction or replication and cultivation of the fungi and virus. Okay, first of all, what is fungi and what is virus? Fungi is a one type of kingdom in which the microorganisms are present. They are mostly causing of the diseases. Okay, these fungi causing diseases to the humans and other type of organisms. And what is virus? Virus is a microscopical agent which is mostly consisting of the DNA and RNA, and the virus is an infectious agent. Okay, and classification of the fungus and virus. Examples of the fungus are mostly yeast and hyphae. There are two types of classification. Yeast and hyphae are the two types of fungal classification. And the classification of virus, they are mostly based on the bacteriophages, enveloped, retrovirus, RNA virus and the single strand DNA virus, Ebola virus, herpes virus, humus virus, papilloma and the rabies virus. This is about the viral classification. And how the fungi reproduction is takes place? The fungus is mostly reproduced through the asexual method of reproduction and sexual method of reproduction. The sexual method is mostly through the spores, budding and fragmentation methods, whereas in the sexual method of reproduction in the fungi is through the plasmogamy, karyogamy and the meiosis. This is about the fungi can be reproduced. And how the virus can be replicate? And the replication of the virus is mostly through the following steps like attachment, penetration and uncoating, replication, assembly and viral release. This is about the steps involved in the reproduction of the virus. And the next topic is about the disinfectants. This is about the disinfectant, and we need to study, before going to disinfectant, we need to study about the how we cultivate the fungus and bacteria. The cultivation of the fungi is mostly through the basal medium by using the tissue culture and along with the dextrose sugar and biological safety food. Okay, this is about the how to cultivate the fungi. And what is the cultivation of the virus? The cultivation of the virus is mostly through the cultured cells, laboratory animals and the natural host cells along with the embryonated hen's eggs. By using the hen's eggs we can cultivate the virus. Okay, next, coming to the disinfectants. What are disinfectants? Disinfectants are the chemical agents or the substances. These are mostly used to kill the growth of the bacteria or germs. Okay, this is the disinfectants are the type of heat radiation. Okay, and then we are going to the classification of disinfectants. The disinfectants are mostly classified as a bactericidal, fungicidal, virucidal, sporicidal, etc. These are the classification of the disinfectants. Okay, next, factors influencing the process of disinfectants. What are the factors? They are mostly affects the process of disinfection. The factors are mostly pH. pH changes can affect the disinfection, temperature, relative humidity, and water hardness. These are the different types of factors. They are mostly affect the process of disinfection. And the next one, antiseptics. What are antiseptics? The antiseptics are the types of chemical agents. They are mostly used to slow or stop or kill the growth of the microorganisms. They prevent the process of sepsis. Sepsis means it is a type of infection which is mostly stopped by using the antiseptics, means they will be opposite the action of the septics. And coming to the evaluation techniques involved in the antiseptics are mostly in vitro test, in vivo test, dilution assays along with the biocompatibility index. These are the three steps mostly involved in the evaluation of the antiseptics. Next, what are bacteriostatic and what are bactericidal actions? The bacteriostatic is a type of bacterial activity in which static means they stop or inhibit the growth, whereas in bactericidal means killing. They are mostly prevent the growth of the bacteria or killing itself the growth of the bacteria. So these are called bactericidal in actions. Next, how we evaluate the bactericidal and bacteriostatic action? The bactericidal activity is mostly done by the using of the two chemicals, that is minimum bactericidal concentration and minimum inhibitory concentration. We can easily evaluate the bactericidal activity, and the ratio is mostly less than or equal to 4, in which the cidal action will be occur, and the ratio is greater than 4 in which the static action of the bacteria will be occur. And most evaluation method is by using of the microdilution assay. The microdilution assay is mostly used to evaluate the bactericidal and bacteriostatic activity. Okay, next, what are sterility testing? The sterility testing is a testing of the microorganisms which is mostly used to find out whether the microorganisms are present or not. So this is about the sterility testing. We are mostly study the viable microorganisms only in this sterility testing of the products like solids, liquids, ophthalmics and other sterile products according to the Indian Pharmacopoeia, British Pharmacopoeia and the United States of Pharmacopoeia. So what are the sterility methods we need to follow for the sterilization of the solids? They are mostly autoclave, dry heat sterilization, flame, alcohols using and ethylene dioxide using. And what are the sterility methods in the liquids? In the liquids we need to use mostly the liquid chemical test along with the ethylene oxide and hydrogen peroxide, bleaching agents and steam. And what are the methods we need to follow for the ophthalmic sterilization? The ophthalmics are mostly sterilized by using the autoclaving, filters and other aseptic compounds along with the bulk heaters. Okay, this is about the sterilization of ophthalmics. And other sterile products. The other sterile products, like what are the sterile products used in the pharmaceuticals? They are mostly ampoules, vials, bottles, bags, tubes and eye drops, ear drops, etc. These are comes under the category of sterile products. These products are also sterilized by using the autoclave and other sterilization methods. Okay, this is about the complete topics covered under the unit 3, and then we will go for the unit 4. The topics covered under the unit 4. The first one, designing of the aseptic area. First we need to know what is the aseptic area. Okay, aseptic area, which is an area in which aseptic means prevents the process of septic means other infection. It is in a sterile area or the sterile environment or the clean room to prevent the contamination. And how we design these aseptic area. Okay, how do we design? The designing is the best method to prevent any product from the contamination. Okay, designing is very important in aseptic. Okay, first the designing consisting of mostly of the construction. The construction involves of using of the different types of floors, walls, ceilings and HEPA filters along with the air filters and windows, doors, staff, etc. This is coming about the designing of an aseptic area. What is the laminar airflow equipments? The laminar airflow equipment is a type of equipment which is mostly used to sterilize the environmental air. Okay, this is about the laminar airflow. And next, what are the different sources of contamination? The sources of contamination is mostly through the many number of sources are there in the aseptic area. The first one is personal. The personal care is very important whenever the person is entering into the aseptic area. Okay, and the next one, raw materials. The raw materials when we are using for the manufacturing or any synthesis of any material, they must be sterile before entering into the aseptic area. Okay, and the next one, working environment. The working environment is also must be sterile, means prevention of any other microorganisms. And the storage facility area is also very sterile, need to be prevent the contamination. And handling also very important in a important in the aseptic area. And the next one is improper labels. The improper labels are also leads to the process of contamination. And then chemicals using. The chemicals using are also one of the source of the aseptic area contamination. Okay, this is about the different types of contaminations mostly occurred in the aseptic area. Okay, next, what is clean area classification? Clean area is also aseptic area. Okay, next, the remaining topics in the fourth unit are principles and methods of different microbiological assay. What is a microbiological assay? The microbiological assay is a type of assay which is mostly used to find out the microorganisms potency, their quality and their action. Okay, this is about the microbiological assay. Okay, and the microbiological assay is mostly deals with the study of preparations under the assay categories. Okay, they are mostly of different types of microbiological assays. They are using of the concentration and potency of the bacteria. How many types of microbiological assays are there? They are mostly dilution methods, turbidimetric method, titrometric method, gravimetric method and diffusion methods. These are the different types of microbiological assays. Okay, and then what is the principle of microbiological assay? The principle of microbiological assay is mostly used to compare or inhibition of the growth of the microorganisms. Okay, we need to find out the growth of the microorganisms. Next, the second topic is about the antibiotics. What are called antibiotics? The antibiotics are the type of medicinal compounds. They are mostly used to prevent the bacterial infections. Okay, these are called antibiotics. Okay, and what are called vitamins and what are called amino acids? The vitamins are small compounds. They are mostly used in the body to function and to maintain the body healthy and good. Next, what are called amino acids? Amino acids are nothing but they are building blocks of proteins. Okay, the group of proteins are amino acids. Next, what are the assessments we need to find out for the new antibiotics along with the antimicrobial activity. Okay, the assessments for the new antibiotics, they are mostly the laboratory conditions like agar dilutions, disk diffusion method, microbiological assay, flow cytofluorimetric method. So these are the different types of assessments we need to follow for the new antibiotics. Okay, next, what are called antimicrobials? The antimicrobials are the substances, they are mostly stop the growth of the bacteria. Okay, next, the last topic in the fourth unit is what are the general aspects in the environmental cleanliness. Okay, and how to identify the antimicrobial activity. The antimicrobial activity is mostly identified by using the molecular test and microbial inhibitory concentration and antibacterial susceptibility test along with the bioluminescence assay exam. Okay, this is about the how to identify the antibacterial activity. And next, what are the aseptic environmental cleanliness? The aseptic environmental cleanliness is mostly used to create and maintaining of the sterile environment in which we need to minimize the process of contamination. Okay, this is about the topics under the fourth unit. And the fifth unit topics are mostly types of spoilage. Okay, first of all, what is spoilage? The spoilage means when a substance is changes its property, means we cannot use it for the consumption or other any activities. Okay, that is called the spoilage. How many types of spoilages are there? And the types of spoilage is mostly of bacterial spoilage, chemical spoilage, physical spoilage and mechanical spoilage. Okay, and factors for that spoilage are mostly bacterial, yeast, mold, spoil, etc. These are the different types of spoilage factors. And other factors like a chemical spoilage, physical spoilage and microbial spoilage of pharmaceutical products. Okay, next, what are microbial contaminants? Okay, the microbial contaminants are mostly the types of organisms like bacteria, yeast, fungi, virus, prostate and protozoa and parasites. These are the different microbial contaminants. And what is the source of microbial contaminants? The source of microbial contaminants like food, soil, water, human activities, pesticides along with raw materials. These are the sources of microbial contaminants. Next, how do we preserve the pharmaceutical products? Okay, we preserve the pharmaceutical products by using the antimicrobial agents. These antimicrobial agents, they are mostly used to kill the growth of the microbes. Okay, and they are mostly consisting of the microbial agents. How we evaluate the microbial stability of formulations? We can evaluate the microbial ability by using the stability methods like microbial limit test, preservative efficacy test, antimicrobial effective testing. So these are the different steps to involve the evaluation of microbial stability. Okay, next topic is what is the growth of animal cells in culture? What is the animal cells in culture? The animal growth of is mostly determined by using of the growth phases like log phase, lag phase, stationary phase. So this is about the growth of animals in the cell culture. What is the general principle involved in the procedure of the cell culture? Okay, the general principle involved in the cell culture is mostly of cell culture involves first of all cryopreservation, that is also called as a thawing. Okay, thawing means we need to store the cells or tissues or other organisms in the cryopreservation conditions. Okay, means less than minus degrees of temperature to sterile the products. We need to preserve them for the long time usage. And the second one is trypsinizing. Trypsinizing means by using of the trypsin enzyme. And the third step involved in the cell culture is cell counting. And the fourth one is a cell harvesting by using of the centrifugation process and cell separation. After that, cell observation. These are the steps involved in the procedure for the cell culture. Okay. Next, primary established and transformed cell culture. After that, we need to transform the cell cultures into the large media. What are the applications of cell culture in pharmaceutical industry and research? How we use the cell culture in pharmaceutical industry and for the research purpose? They are mostly used for the drug discovery and the drug development, along with the vaccines production and toxicity testing studies, along with the manufacturing of biopharmaceuticals and medicines, regenerative therapy, and drug permeability studies. This is the what are the applications we need to follow for the pharmaceutical industry by using the cell culture. So this is about the topics covered under the fifth unit. So that's all for this session. Thank you, and please do subscribe Pharma